This is a production of Cornell University.
Okay, hi everyone. So I'm Jimei Han. So I'm very glad to have this opportunity to present my research here. So today I'm going to talk about how to mechanistically estimate photosynthesis and photosynthetic capacity from solar-induced chlorophyll fluorescence by utilizing the physiological foundations. So first, I'd like to talk about the motivations of this research. We know photosynthesis is a composed light reaction and a carbon reaction. During the light reaction, the energy from sunlight absorbed by the leaves is dissipated by three pathways. So the first pathway is to enter the electron transport chain and then produce the reduced energy ATP and ADPH, which can drive the carbon-benzene cycle in the carbon reactions. And the second dissipation pathway is to dissipate the energy as heat, or known as non-photochemical quenching, abbreviated as NPQ. So the third pathway is to emit the energy as the chlorophyll fluorescence. So during the whole process of light and carbon reactions, SIF is the only optically detectable photosynthetic signal. It's tightly coupled to electron transport, which drives the carbon reactions. So therefore, the rapidly growing cross-scale observations of SIF can offer us a promising opportunity to quantify photosynthesis across different spatial temporal scales. So with the emerging interest in taking advantage of information carried in SIF to constrain photosynthesis by utilizing the balance between light and carbon reactions, it's critical to develop an approach that enable direct and independent estimates of photosynthesis. So the initial findings have identified the linkage between SIF and photosynthesis. So the first from the observational side, the existing studies, they have found the empirical linkage between SIF and photosynthesis inferred with the eddy covariance mechanism of net ecosystem exchange. Photosynthesis inferred with this way, although often assumed as truth, is not directly measured, but is indirectly and imprecisely derived with approaches known to contain errors or even bias. So the potentially biased photosynthesis used as truth to infer photosynthesis-SIF relationship essentially contradicts our motivation in applying SIF to constrain photosynthesis. And secondly, from the modeling side, the existing studies primarily adopted the scope model to simulate SIF photosynthesis relationship. Specifically, the scope model first utilizes the FVCB biochemical model to calculate the photosynthesis and then photochemical quantum yield, which in turn is used to calculate the chlorophyll fluorescence yield and then SIF by empirically linking non-photochemical quenching models. So while useful for simulating SIF photosynthesis relationship, but the scope-based strategy cannot escape the well-known uncertainties from the parameters and structures. For example, the direct use of leaf levels FVCB biochemical model at the canopy scales will cause the uncertainties at the larger scales. Another uncertainty source is from the empirical NPQ model. We know NPQ is a non-photochemical quenching is a very complex parameters. It contains multiple components, but here this empirical model only consider NPQ as a single parameters, so that it will constitute the uncertainties. So also it's circular to use the establish the SIF photosynthesis relationship to back calculate the photosynthesis. So based on these uncertainties from both observational and the modeling side, we were thinking about how should we utilize the mechanistic information content in observed SIF to estimate photosynthesis independent from the existing photosynthesis estimation approaches that are known to have different levels of uncertainties. So here we have a mechanistic light reaction model with SIF. So this model is established from a theoretical relationship between SIF and photosynthesis with the actual electron transport rate as a bridge. Specifically, this model first utilizing SIF to calculate the actual electron transport rate with the QL-based equations. Here, QL is the fraction of open reaction centers. We can also use the NPQ-based formulations to calculate the actual electron transport rate. So these two equations are mechanistic equivalent, but here we only choose QL-based equations. That is because although these two equations are theoretical equivalent, but practically they have different complexities. Like the QL, it reflects the redox state of PS2, and it provides the steady state approximation of the steady the cytochrome B6F that plays a central role in the steady photosynthesis. But NPQ, it involves both energetic and enzymatic reactions. Although the heat dissipation from NPQ is predominantly localized in the complex, in the light harvesting complex, its activation and regulations occurs in the lumen, in the lumen and thylakoid membrane and stroma. And also NPQ has a delayed response to the light variations. It also contains multiple components. Each component operates at different time scales. So this complex activation, regulation, and the delayed time response and the involvement of the multiple responses in the dynamic of NPQ components, increasing the complexities in modeling NPQ as compared to QL. So once the actual electron transport rate is calculated based on the QL equations, then the photosynthesis can be directly calculated with these equations. So from this scheme, we can see this model has a clear separation between SIF and photosynthesis. It can avoid the circularity issues we mentioned above. To verify these models, we first focused on the leaf scales and collected concurrent measurements of gas exchange and chlorophyll fluorescence from the variety of species. From the 11 plant functional types native to the temperate, tropical, and boreal climates in the Cornell Botanical Gardens, the tropical botanical gardens, and also the working in growth chambers at Oak Ridge National Lab. Here we thank for the Yung Zhang and his students from the University of Mayo for collecting the data in tropical species. Utilizing this wide, here is the pictures for our measurements in the Cornell Botanical Gardens. So utilizing this wide range of species, we first tested the relationship between SIF and the photosynthesis. We find a nonlinear relationship between SIF and the photosynthesis across different plant species. This nonlinear relationship is characterized by the initial increases of photosynthesis and then leveling off when the light intensity is high. So this nonlinear relationship is because the photosynthesis saturates at high light, but the SIF keep increasing at high light. And also we find a different degree of nonlinearity between carbon-3 and carbon-4 species. This nonlinear relationship can be explained by our formulations. So here we can see the QL, the involvement of QL will prevent the dependence of photosynthesis on SIF. we then tested this relationship under different temperatures. So we find the similar results and also different level of the saturations and the rate approaching the saturation level among carbon-3 and carbon-4 species. So this nonlinear relationship is not consistent with the patterns that at the canopy level that reported a linear relationship between SIF and photosynthesis. That is because at the canopy level and beyond, the canopy structures or the leaf error index dominate the relationship between SIF and photosynthesis. But this linear relationship tends to break down at a shorter time scales. For example, recent studies, they find a decoupling between SIF and photosynthesis when artificially closing the stomatal conductance by adding the abscisic acid hormone to so the closed stomatal conductance will decrease the intercellular CO2 concentrations and then decrease the photosynthesis. That caused the decreased demand for the reduced energy ATP and NADPH. So to keep the balance between light and carbon reactions, plants will decrease the QL and or increase NPQ to dissipate the excess energy to decrease the production of reduced energy. And then we tested the performance of our model. So this figure can show that our model can reproduce the measured photosynthesis very well across different plant functional type species. And also we tested its performance under different temperatures. Its performance still very well. Then we tested the uncertainties of our model. So before showing the results, I'd like to strengthen one point, and that is our model and the traditional used FVCB biochemical model of photosynthesis at the leaf scale is equivalent. So the equivalent lies in the balance between light and carbon reactions. So the difference between these two models is that our model is from the perspective of light reactions, but the traditional used FVCB biochemical model is from the perspective of carbon reactions. And another difference is that our model can be used only if SIF is available. So based on the equivalence, we tested the uncertainties of these two models. First, by utilizing the TFD specific parameters, we compared the performance of these two models. So it shows our model performs slightly better than the FVCB biochemical models. So the FVCB biochemical models slightly overestimate the photosynthesis. And also we using Monte Carlo simulations to test the uncertainties of these two models. So the figure A shows that our model suffers less uncertainties at different light intensities, and also shows a narrower density distribution of photosynthesis at a saturating light. Here, the performance difference is not due to the model rigorousness, but due to differences in the level of parameter requirements. Specifically, our model takes advantage of the information carried in SIF and then shortcuts the uncertainties propagated from the imperfect model parameterizations. Again, here we compare these two models. It's not because we want to show which model is better than the other or which model has a weakness, but we only want to show that our model takes advantage of information carried in SIF, so that it will decrease the uncertainties of photosynthesis estimations. So to further facilitate our model to a larger scales, we still have some have more works that need to be done in the future. So first, we need to couple a leaf radiative transfer scheme or invoke escape probability factors to infer the total sieve from PS2 from the remotely sensed sieve. Or we need to we need to mechanistic model the response of QL under environmental conditions. And then exactly estimate the KDF parameters. That requires a development leaf-level for spectral fluorescence systems that enable separating PS1 and PS2 contributions. And eventually, we also need to couple the stomatal conductance model and our model. So that is much like the classical FVCB biochemical model is applied in a terrestrial biosphere models, or the land sphere, or the land surface models. So here is a simple summary for the above research. So that we know in the application of the FVCB biochemical models at the larger scales, the parameters like the Vcmax, Gmax is the maximum carbon oxidation rate or the maximum electron transport rate is often assigned a specific values for each plant functional types. So that is uncertainties will constitute the major source of prediction errors in the simulating photosynthesis at the larger scales. We know these two parameters are very important and it can determine photosynthetic rate and its response to environmental changes, so that the timely and accurate estimation of these parameters is important. So currently, the sieve emerges as a promising remote sensing tool to infer Vcmax and Gmax. So we were thinking about how should we utilizing the sieve to mechanistically modeling photosynthetic capacities in natural environments. So the current studies have found the mixed results for the relationship between sieve and Vcmax. For example, there's one study. There are few studies. They first set the Vcmax to a range of realistic values, and then using the scoop model to simulate the sieve Vcmax relationship, and they find a linear relationship, the positive linear relationship between Vcmax and the sieve in a fixed day. And there's another studies. They find no relationship between sieve and Vcmax or Gmax, but find a negative relationship between Vcmax, Gmax, and sieve yield. So here sieve yield is calculated by the sieve divided by observed light intensities. So the negative relationship between Vcmax sieve with sieve yield is attributed to the linear positive relationship between Gmax, Vcmax, and the observed light intensities. So both the observational results and the simulation results, they didn't consider a mismatch in relating the Vcmax to sieve. Specifically, we know the Vcmax and the Gmax calculate the photosynthetic capacities, and they suppose two parameters that do not depend on the fluctuating light, but the sieve is emitted only from the light reactions. They vary rapidly with the fluctuating light in the natural environments, like the sieve increasing with the light intensities, and also it has a diurnal changes. So to better understand the causes of the discrepancies of the existing studies and develop a mechanistic solution to infer Vcmax and Gmax using sieve, we first sought to establish a theoretical basis that links the Vcmax, Gmax, and SIF by balancing the actual electron transport rate derived by the FVCB biochemical model, and that derived by our mechanistic light reaction model. So first, under the Lubitsch limitation status, we can derive the functional relationship of Vcmax with SIF. So here we only derived Vcmax equations, but a similar relationship of Gmax with SIF is also expected. That is because Vcmax and Gmax has a close relationships among different species and environments. Under the RUBP regeneration limitation status, we also derived a functional relationship of Gmax with SIF. A Vcmax relationship is also expected under this limitation status. So in these two equations, we can get two hypotheses. So the first hypothesis is that the SIF alone cannot be used to infer Vcmax and or Gmax, but the products of QL and SIF can be better used to infer Vcmax and Gmax. The hypothesis 2 is that Vcmax and Gmax have different relationships with SIF times QL between Lubitsch limited and RUBP regeneration limited carbon oxidation states. That is because in the formulation of Vcmax with SIF, we can see the slope. The parameters in this slope are often assumed conservative, but in the formulation under RUBP regeneration status, the slopes in contains QL, SIF, and power variables, and it will impact the direct dependence of Gmax or Vcmax on SIF times QL. To verify these two hypotheses, we further fitted Vcmax and Gmax with the traditional approaches, that is fitting CO2 response curves based on FVCB biochemical models. Here we didn't use our theoretical equations to calculate the Vcmax and the Gmax. That is because the parameters in these formulations are often assumed as constants, but they are currently unknown. So these assumed constant values will cause the inaccurate Vcmax and Gmax values. First, we used the species from 6 plant functional types. We find that there is no relationship between SIF and Gmax, Vcmax. But if QL is accounted for, then there are a significant relationship between SIF times QL with Gmax and Vcmax. And also QL is significantly related to Vcmax and Gmax. But compared with the SIF times QL, the explanatory power of QL for Vcmax and Gmax is reduced by 11% or 30%. So that indicates the SIF times QL can be used better in for Vcmax and Gmax. We further tested this relationship under different temperatures. We find the similar results, so that these results can confirm our hypothesis 1. So our theoretical and measurement results consistently show that there's no general or predictable relationship between SIF alone with Gmax and Vcmax. And our study focused on the leaf level. So even at the leaf level, the relationship is already complexes. At the canopy level, there are additional complexes, such as the structure, the canopy structure, and the reabsorptions of SIF within the canopies, or the vertical variation of QL will impact the strength of the relationship between SIF alone and Vcmax, Gmax. And also from our theoretical equations, it's mechanistically unwarranted to expect a simple relationship between Gmax, Vcmax, and SIF alone. So this can explain the conflicting results in existing studies. Another findings is that the close relationship between Gmax, Vcmax with SIF hold relatively stable among temperatures. So to understand it, we compared the temperature dependence of Vcmax, Gmax, QL, SIF. So we find the Vcmax, Gmax, and QL has a similar patterns with the temperatures. But the temperature has a slight has a weaker impact on SIF. So the Vcmax, Gmax, QL they increase in parallel as function of temperatures from the 20 to 35 degrees, and then decreases at a higher temperatures. And also we find a linear relationship between Vcmax and QL. So that might suggest the QL can can reflect the activation of carbon oxidation enzymes. Then we further tested the hypothesis 2. We find the linear relationship is stronger under Lubisco limited than under IOBP regeneration limited status. That suggests that when SIF times QL is used to infer Vcmax and Gmax, we need a priori information of which carbon oxidation limitation status SIF is under. So here we can use the response of actual electron transport rate to light intensity to identify the limitation status. So for example, if the actual electron transport rate is increasing with PAR, then the data belong to the IOBP regeneration limitation status. But if the actual electron transport rate plans plateau with PAR, then the data is Lubisco limited. So here, interestingly, we find the SIF times QL has a similar pattern with the actual electron transport rate. So that means if the QL is mechanistically modeled, then the SIF times QL at the canopy level and beyond, it can be used to identify the carbon oxidation limitation status. So here is the similar patterns can be also explained by our theoretical equations. So that means the QL times QL can be directly used to calculate the actual electron transport rate. Here is the brief summary. So first, the SIF alone is incapable of informing the evaluations in Vcmax. Even when SIF is determined under the same environmental conditions. Secondly, our findings suggest that the redox state of PS2 reflected by QL strongly influences the relationship between SIF and Vcmax Gmax. The limitation state of carbon oxidation must be considered if observed SIF is utilized to infer Vcmax under dynamic environmental conditions. So here we thank for the contributors on our research. Thank you, that's it.
Yeah, thank you very much, Jimei. Any questions from the audience? Feel free to unmute yourself and ask questions directly or post your question to the chat window.
Well, Ying, this is not my area at all, but so I was looking, it seemed like in a lot of the relations I saw at the higher concentrations of either SIF or assimilation or whatever, you had much larger spreads than at the lower ones. It seemed to be a common thing in a lot of these scatter plots. Was I seeing that correctly, Jimei? Oh, and what explains that? I don't know which slide. You had a lot of slides where you're you're showing this relation, and at the higher levels, there would seem to be much more spread, a lot of little gray dots. And when you got down to lower concentrations, you had much less error. It seemed to be a kind of this funnel shape. I don't know if I saw that correctly.
Yes, that's a good question. Yeah, our study focuses on the leaf scale, but we know only at the leaf scale, the functional process can be fully investigated, and even at the leaf scale, we know maybe the relationship is already complexes, or so. I mean, at the chemical level, except the physiological factors, there are additional complexities.
Yeah.
So it will affect the relationship.
Yeah.
So
okay,
it
that
will,
wasn't really my question, but that's okay. I guess I didn't make it. I didn't make it clear enough. Feel some of your scatterpoints. Yeah. Tim, you probably can see that better than I can, so I'll let you go ahead. I'll let the real plant physiologist talk here.
Well, yeah, but actually my question is very simple, and I just was going to ask you to maybe tell us a little bit more background on what is QL actually evaluating. You say that it is the redox state of photosystem 2. So could you just spend a little bit of time to explain further what is QL telling us? What is QL?
Yes, QL is the fraction of open reaction centers in PS2. Yeah, it can reflect the redox state of PS2, and also it reflects the it can varies with both light and carbon reactions. I mean. It can reflect the balance between light and carbon reactions.
Okay. So what's in a very simple way, it sounds like QL is helpful in telling us whether we are in a light limited situation or a carboxylation and CO2 limited situation. Is that correct? Maybe not.
Yeah, yeah, Tim, I think that is quite accurate summary. QL, so I mean, the take-home message in the second part is that SIF itself is at least from at very fine scale, SIF itself is not sufficient to infer photosynthesis or Vcmax, these key parameters. But if we can borrow the information content in QL, then we can do a much better job. So basically the carboxylation limiting state, whether it's Rubisco limited or light limited, that information is implicitly contained in QL. And that information should also be to some degree contained in SIF, but again, not enough to directly infer the Vcmax. Is that clear?
Yeah, that's great. Yeah, that sounds great. Thank you.
Yeah, thank you. That's a really great question. Thank you. David, I would like to share my thoughts of to answer your questions. I think your observation is absolutely correct. It's just like at the high end, there are more environmental factors that work together to influence their relationships. But at the low end, it is typically dominated by a single environmental factors. So that means the relationship is going to be more complex at the higher end. That's why you see more scatters, because we can only show the relationship in 2D plot. That's why the influence from other factors is just basically hidden there and create more scatters or more spread. Does that make sense?
That makes perfect sense. I was thinking of something like that. Yeah, because at the lower, it's only dominated by one process. Yeah, I know
Yeah.
there were quite some graphs like that. Thanks. And then
Okay.
you wonder, you just wonder if that the other, just statistically speaking, when you look at that 1-to-1 line, yeah, you're fitting it, but it's not uncertain, the same uncertainty throughout the whole line. So it's really not exactly correct in explaining. I mean, the overall error is more concentrated in one part of it. That's all. So
Yeah. It is very complex. So that's why we have to consider additional factors. We do have like a 3D plot. Not sure if Jimei has shown that before in her slides, but yeah, we do want to highlight the complexity. For some reason, reviewers do not like the 3D map, so we have to convert to 2D. Yeah.
okay, that's clear. Thanks a lot.
Yeah, thank you. Any questions? If no other questions, thank you very much, Jimei, for this great presentation. We'll hope Jimei will have a successful career in the future once she returns to China. Yeah, thank you.
This has been a production of Cornell University, on the web at cornell.edu.